Greetings all, and welcome to another episode of Secrets from the Crime Lab, an educational podcast where we inform and encourage discussion on forensic science and related scientific fields and topics. I'm your host, Angela Swartz. I have three guests with me today, which I will introduce to everyone shortly. But first, I wanted to do a short little announcement for everyone. I did complete season 1, and so this is one of 2 bonus episodes that we're going to have. We had some scheduling difficulties related to people like myself having the flu and other people having COVID, so things have been a little bit delayed. But so we're going to have 2 bonus episodes for season 1 that I'll have out for you. I'm scheduling guests for season 2 at the moment, so that will be sometime this fall when those episodes will be available. In the meantime, I wanted to thank everyone for tuning in and listening. We've already crossed the threshold of 1,000 downloads, and I really haven't done any marketing, so that's really amazing for all of the support, and I couldn't have done that without everyone sharing this around with their friends, so I really appreciate that. I have had some requests to do video content to go along with the podcast. At the moment, this podcast is completely self-funded. So if video content is something that you would like to see in the future, I would appreciate your support to get there. If you would consider making a donation to help cover production costs, if you're listening on a podcast app, there's a little link at the end of the show notes that says support the show. That'll take you to some place where you can make a donation in whatever quantity you want. Currently, I'm using Buy Me a Coffee, so you can literally buy me a coffee, but I might change that to Buy Me a Taco since I'm moving back to Texas. And if you're listening on the web player, there's a little heart icon next to the social media links, and you can just click on the heart and it will take you to the same place. Okay, and so with the announcements out of the way, let's move on to our topic for today. So I'm doing this in partnership with the Association of Forensic DNA Analysts and Administrators. Their annual summer meeting is taking place in Houston at the Royal Sonesta Hotel, July 28th and 29th, I believe. I'll put a link in the show notes because I may have those dates incorrect for anyone who's interested in attending that. So the day before the meeting, there is a free workshop on the analysis and validation of forensic MPS data. And MPS is massively parallel sequencing, and that is what we're going to be talking about today. That workshop is hosted by Soft Genetics, and it's Wednesday, July 27th from 1 to 5 p.m. And so we're going to just talk a little bit about what that workshop is going to be about. It is going to be a pretty technical workshop, but it is open to anyone who wants to go. So whether or not you're a scientist, if you're a prosecutor, defense, or in the Houston area and really interested in that sort of thing, you can certainly RSVP for it. But it is going to be technical, so you are forewarned on that. So massively parallel sequencing is something that generates a lot of data, and that's something that we're going to talk about later on in the podcast. Soft Genetics is a company that I'm going to allow Teresa to talk to you a little bit about what their company does. So they do software solutions for data analysis. And I also have her colleague, Sarah, with us today as well. And then we have Megan, who's a visiting professor at George Washington University at the moment. And I'm going to hand it over to Megan to start with and let her introduce herself to everyone.
All right. So hello. Thank you for having me. So yeah, I can talk a little bit about how I got to where I am today. So I have a Bachelor of Arts in Biology from the College of St. Benedict, which is a small private school in Minnesota, which is where my home is. And then I went and got my master's degree in forensic science at Arcadia University. So at that time, after my master's, I went and started working in a private crime lab in the area doing forensic biology casework. So I did a lot of different types of casework from pretty much start to finish, from opening a case to reporting. And then I also got into a lot of the validation work, so testing the new products that are coming out in the field and getting them ready to run casework on. And so validation, if you've ever done it, it's kind of on the research side of things. I love it. Not a lot of people will tell you that. I do love validations. But because of that, I actually decided to go into more the research side of forensics. So I switched to a different department at the company, focusing on training analysts, performing research, and then the education part. So during my time there, I decided I want to go back and get my PhD. So I'm working on that right now through Oklahoma State University in forensic science. And then I took a job here at George Washington University in DC as a visiting professor, teaching for their master's program in forensic science. So a lot of the research I've done, because I have bridged both worlds of casework and research, I can see it from a different perspective of how do you implement the research that's going on. There is some awesome research going on by institutions and universities, but how do we get it into the crime lab then? Because there's a lot that goes in between first finding out about it, but then getting it ready to go to court. So I have focused a lot of my research on whether it's new products or current products, testing the limits out, and finding ways that labs can take the research I've done and use it to implement in the lab. Tell them you should consider getting this instrument or this piece of machinery, or consider this when you're validating it. So a lot of it could be a new product. A lot of what I'm focusing now is on the implementation of MPS. So it's a whole new realm of data and statistics. So that's really what I have been doing right now.
Thank you. Thank you for coming on the podcast.
Absolutely.
I appreciate it. Sarah, you want to go next? Introduce yourself.
So let's see. I got my bachelor's in physiology, actually, at San Francisco State University. I took a little break in between and before I got my master's, and I did that at University of California at Davis, and that was in forensic science, focusing on DNA analysis. And I did my thesis work, NGS analysis of degraded bone samples. After that, I started working at Mitotyping Technologies for a few years as a forensic DNA analyst. And so mitotyping specifically does mitochondrial DNA work. So that's mainly hair shafts, things like that. And so I worked on that from extraction through the whole process to writing the reports and testifying in court. After that, I moved on to soft genetics, and here I get to help people troubleshoot, help them learn how our software and things like that. So it's a lot of...
So you do a lot of training and education for people that they're using the software in their day-to-day work. Yeah.
I do a lot of the training so that they can use it day to day. If they get difficult samples that they're unsure of what to make of them, I work with them to figure that out. So it's always interesting.
So you're the phone a friend.
Yes.
You're the phone a friend for the...
Yes.
Is that TV show even still on anymore? I don't even remember what it was called, with the phone a friend thing.
Millionaire, yep.
Who wants to be a millionaire? Yeah.
Right. Yes, I think all of us would like to be a millionaire. All right, so Teresa?
Yes, thanks so much for having us on your podcast. And I started my academic career completely different area, ecology and environmental science for my bachelor's degree, and then worked as technician, various positions. By the time I got to Penn State, I switched into molecular biology of disease resistance. And so that involved developing PCR primers. We did a lot of Sanger sequencing and a lot of basically gene discovery in disease resistance in agriculture. And then for about 15 years, I was a biology and biotechnology professor up in New York State. We moved back to Pennsylvania and switched gears and have been working for soft genetics for about the last 15 years, where we develop forensic DNA analysis software for any of the different platforms. There's another side of the aisle where we also are very active with basic research software. So any type of MPS research, but that's separate from the forensic because the forensic has specific needs. In our software development, as Sarah was saying, we do a lot of training, support, assistance, and interaction with the labs that are using our software. So helping them to get the most out of the software and also getting suggestions for them of what functions would be helpful to include in new versions. So it's a lot of teaching, but not in that university lecture hall lab type of setting. It's much more one-on-one or one to small group, and in very specific areas, depending on where the laboratory has, what their research or what their lab and data analysis requirements are.
So it's professional, on-the-job type trainings for specific applications, whether it's in a research laboratory or in a production forensic laboratory.
Absolutely. Yeah. Unlike the freshman classes, no one asks me if they have to learn this or will it be on the test. It's very refreshing to interact with professionals who are very motivated to become skilled and understand what they're working with.
Yeah.
Stay with us. We'll be right back.
Hey there, everyone, and thank you again for listening to an episode of Secrets from the Crime Lab. I wanted to take a moment to ask you for your support, because if you're like me, you're not a fan of ads interrupting your shows. But in order for Secrets from the Crime Lab to remain ad-free, I need your help. So if you would like to support the show, there are 3 different ways that you can help the show right now. First, and perhaps the simplest thing that you can do, is just to share it with your friends. Get the word out. That will help a lot. Share our little science corner of the universe. The second thing you could do is maybe consider making a one-time donation. Or, the third thing, become a paid subscriber, and that starts at as little as $3 a month. It's super easy to do. The links are in the show notes and whatever podcast app you're using to listen to the show, or you can go directly to secretsfromthecrimelab.com and click on the little support button with the heart on it. Every little bit helps to cover the production costs, and thank you again for listening, and I hope you learned something new today. All right, in everyone's introductions, we threw out a lot of jargon, like DNA nerd jargon that not everyone that listens is going to know what it is. So we've said MPS, we've said NGS. Does anyone want to explain what those are?
Basically, NGS and MPS are 2 different abbreviations for the same thing. It was originally, I think, called NGS, which stood for Next Generation Sequencing. But it's now been around for 15 or more years, so it's not really a good name for it. MPS is much more descriptive. It stands for Massively Parallel Sequencing. And so that describes what's going on, is that you've got many different sequencing events going on all at the same time. And so you're gathering this depth of sequence information that prior to this technology being developed was not possible to have that depth of information. And actually, historically, the development of MPS is why the Human Genome Project, that probably people remember back when we were on, scientific community was undertaking sequencing the human genome, it actually finished ahead of schedule because they originally thought they would have to use Sanger sequencing for the whole thing. MPS was developed, and so that kicked everything into high gear.
And so we're talking about 2 different types of sequencing. So we say the old sequencing was Sanger-type sequencing. Sanger was a person's name that developed the technique. And that's where we're just looking at a single stretch on one piece of DNA, basically, to start with. And we're looking at that individual sequence of that small section. Back before we even had automated, we were doing manual sequencing where we were even using radioactive materials to label it and stuff. So we would get short stretches, 300 to 600 bases. But when we're talking about massively parallel sequencing, we were saying it's massive and in parallel because we're sequencing all of these different stretches all at the same time. Just in multiple samples at the same time. Yes, the throwing all of your sample into one bin with biochemical barcodes on them. It's definitely a paradigm shift for those of us that work in a forensic laboratory where we're like, no, everything has to be kept separate. You can't do that. And I know some of this is going to be, even though we're trying to keep this as a high level discussion, I know that some of the technical side of things, it's hard for us not to get a little bit technical in some of the conversation. But the MPS was a massive leap forward technologically for anybody who was doing any kind of DNA work, right? Any kind of research, like you used to do agriculture research. I used to do cancer research on specific tumor genes before I went into forensics. And that's one of the things that those of us who are interested in forensics or in the forensic field sometimes forget, because we get very focused on forensics. But all of these new technologies mostly developed in research and clinical side. And then forensics goes and looks at it and goes, hey, this is a really neat technology. We can do a lot with this. Let's just tweak the sensitivity because we have degraded samples and low quantity samples. How do we make this technology that you guys have figured out over here on the clinical side? Now let's pull that into forensics now that you've worked all the kinks out of it and make it a little bit more specialized for us. And so that's one of the things that we talk about. There's aspects of this technology that are really helpful for. Sarah mentioned that she worked with bone samples quite a bit and hair shafts, and that's specifically usually mitochondrial DNA. So, Sarah, could you tell us a little bit about why we want to use mitochondrial DNA when we're looking at bones and hair shafts?
Sure. Bones, hair shafts, they don't have a whole lot of nuclear DNA. That's the stuff that comes from your mother and your father and creates who you are. There's a lot of DNA present in your mitochondria, and that there's a lot of mitochondrial DNA, a lot of mitochondria in general in bones. And so it's a lot easier to get that information from those samples rather than the nuclear DNA, which isn't really present.
The mitochondria are present in like hundreds or thousands of copies in a cell, whereas your nuclear DNA, you've only got one set of your nuclear DNA in each one of your cells. But when we're talking about like anthropological samples, like bones that have been dug up, the nuclear DNA is all degraded and gone. So the mitochondrial is the only thing we'd be able to get DNA from usually.
Yeah, exactly.
So we got the mitochondrial applications, and we can use MPS to test the mitochondrial DNA as well. And we can get a lot more information out of that than we could using the older techniques. And I think, Teresa, you shared a paper, it was a review article, where someone had gotten a mitochondrial DNA from like a 1 millimeter of a hair shaft?
Yes. Yeah, it was amazing. And actually, that's one of the presentations that will be at the workshop, is that routinely they're able to get complete mito haplogrouping from 1 to 5 millimeter pieces of hair. And so one of the slides shows like 1 millimeter is basically a pencil point and 5 millimeters is the eraser. But not, as Sarah said, you would not be getting nuclear DNA out of these. There's no root. There's no nuclear DNA. They tested recent samples, I think samples that were 5 or 6 years old, and some that were 40 to 50 years old. So they were still able to get from the 40-year-old samples, which would be in line with the, cold cases, some of the cold cases. So not ancient DNA, but a cold case, that body that's been found after 30 or 40 years, to be able to get that information. And so the mito is going to give them information about the maternal lineage of that person. And if they can sequence the whole mito genome, so about 16,000 or so, well, a little more, base pairs from the whole genome, they can even separate, in I think it was 40% of the cases, they can separate the mother from the child because of mutations that occur. And so for the most part, the mother-child would have the same mito sequence, but when they do the entire sequence, not just the control region, they're more likely to find heteroplasmies or slight differences between the mother and child. And so you can actually, in many cases, even differentiate between a very close, you don't get any closer than mother-child for mito transfer.
That's pretty amazing.
Yeah, so that's the sort of thing that I think 2 centimeters is more commonly used for mito extraction, but they went through the very small pieces, and that's one of the presentations. I was amazed.
That's actually impressive. And I think you're right about the 2 centimeters. I think that's what I've sent off to the laboratories that do that sort of work. So it's got a lot of applications for the missing persons, unidentified human remains, disaster victim identification. So if we have mass disasters, it's certainly going to be helpful because, I mean, you probably wouldn't be using like 1 to 5 millimeter piece of something when you've got those kind of things. You're not necessarily, it's more about matching up the remains to the right person. But it's still amazing that you can do that because normally with the traditional Sanger type sequencing, where you were only able to look at the control region, you wouldn't be able to differentiate between the mother and the child. You need the whole genome to do that. And you can only do that with MPS.
Right. Yeah. Sanger sequencing the whole mitogenome would be way cumbersome. And you only get, as you were saying, 1 copy. So you can miss heteroplasmy and minor differences with Sanger that MPS is able to pick up.
Forensic laboratories, when we look at things, mitochondrial DNA isn't that's not our go-to. That's not the first thing we look at. The first thing we usually look at are our standard, what we call STRs, or short random repeats. And I know, Megan, you said that a lot of your research is you're focusing more on the big picture for all of the data. Could you talk to us a little bit about STRs and also why STRs? What are they and what are we looking at in those, and how the newer technologies like the MPS is so beneficial to forensic laboratories once they have the money to get the equipment?
That is true. Yeah, absolutely. So STRs are parts of the DNA that as humans, we each share on these sections. Now, they're not genes. We don't look at genes in forensics with STRs, but they are just short base pair or building block repeats that might go ATGC as an example and repeat over and over again. And each person might have a slightly different number of how often these repeat. So that's where the whole the short tandem repeat comes in. There are, at least in the US, we focus in on 20 locations of these STR regions. And by gathering all that information, we can start to develop unique DNA profiles that way. So with that is your nuclear DNA. So you do have 2 copies, one from your mom, one from your dad. So at each of these locations, you're going to see 2 separate numbers, alleles. And alleles are just different variations of whatever that gene or STR could be. So this is what we do in typical casework. If we have enough DNA and nuclear DNA, we can create this STR profile and then use this data. It comes out it looks like a graph, comes out with these large peaks, and then there's a number underneath it. We use that number to tell how many repeats there are. So you could have 2 numbers or you could have 1 number, and that just, again, depends on the allele that you inherited. Sometimes it is the same between both parents, and that's called a homozygous location. Heterozygous is when you have 2 different numbers of repeats. That's basic autosomal STRs. We also can use YSTRs as testing as an additional tool. Now, in your chromosomes, you have your sex chromosomes, and for females, you have 2 X chromosomes. A male's going to have an X and Y. So YSTRs are STRs on the Y chromosome. So it's a tool we can use to focus in on if there's a male contributor. And this is really beneficial when we're doing cases where there is a high amount of female and maybe a low amount. Sexual assault cases are very common, and STRs are super useful for that, in that we can kind of almost block out. It's like if you think of a radio and there's multiple frequencies, we can block out the frequency of the female to focus in on that male component of the DNA. So it just lets us just look at that. Now, similar to what you guys talked about with mitochondrial DNA, the YSTR profile is passed on paternally. So you are going to have the same YSTR profile as your father, as your biological brother, as your uncle, you're a father's brother. That it just changes how we will report on the results for that. But moving into the MPS or the sequencing realm with it, right now our technologies will give us that number. It'll say ATGC repeated 15 times. Now, with the sequencing aspect, since we can look at every single little nucleotide, we might find that 2 individuals that both shared, I'm giving as an example, the number 15. Now under sequencing, we might be able to see there's actually a difference between that 15. I use ATGC as an example. One person might have ATGC and one person might have ATCC. During our normal methods, that looks the same. With sequencing, we can actually see the difference between the 2. Much more discriminatory.
I think there was an analogy. I could be incorrect, but I think it was in the book Forensic DNA Typing by Nora Rudin and Keith Inman. They talked about STRs as a train, and everyone has an engine and a caboose, but the number of cars in between changes, right? So the length of the train is the same. Everyone's got the same length of train, but the number of cars in between changes. And then if we're talking about the individual sequencing, we would say that the length of that train is the same, but then we can tell that the upholstery on the inside of one of those cars is a little bit different.
Yeah.
Is that kind of...
Absolutely wonderful.
Yeah.
Yeah. We're talking about the upholstery, but we would also talk about that this one has a seatbelt and this one doesn't have a seatbelt, and this one's got tinted windows and this one doesn't have tinted windows. I mean, that's starting like every little detail you could possibly think of inside of those train cars. We can start to understand and visualize with MPS.
Yes.
That adds a layer of complication to data analysis. And we need software tools and we need researchers to tease through all of that so that we know, what are we doing with all of this information? How do we convey it in a courtroom setting? How do we make it useful for investigative purposes? And I know, Megan, you'd mentioned that part of your research is you've done a survey of crime laboratories and try to see where are the labs at the moment with understanding the technology, acquiring the technology, understanding what to do with all of this information.
Yeah, absolutely. A colleague, Dr. Fabio Aldoni, and he is a professor at Arcadia University. We both have experience in the sequencing realm and thought there is so much. Every lab's working on implementing it in their own way, but where is everybody at and how can we help as on the research side of things? So we did send actually globally a survey out on just current status. Do you have an NGS? But also, if you don't have a sequencing instrument, what are the hurdles? What are the challenges you're dealing with trying to implement it? So a lot of what I'll talk about during the workshop are some of the results we saw on, of course, the biggest hurdle is funding. Unfortunately, I can't do much about that as a researcher because we all could use a little bit more to help. But there were some other responses that a lot of the respondents did say, things like lack of standards and recommendations. And something I'll bring up during the workshop is there's actually some out there. So we're hoping that the results of the survey just help educate people around the US, around the world of you're not alone in this, but also there's resources you can use. And there was a lot of concern about validation. So there are some SWGDAM guidelines, so some guidelines that we often follow in the forensic field. And also a big concern is how are we going to do the statistics? Right now, we as a field are implementing a statistic called probabilistic genotyping. And it's not something we can do by hand. We need the software to do it. And now that we have...
I've done it by hand.
Oh,
I did it for
amazing.
one locus on a validation. I did it by hand.
Yes.
I will never repeat that. It took me like a week. It took me a week.
Yeah, our turnaround times would go way up if we had to do that. Yeah, but so now that we have all of this, we have so much more data now doing the sequencing, the statistics are just going to become more complex. So there's a lot of opportunity to bridge that gap and to get these two different techniques to come together so we can actually do casework on sequencing data.
One of the things that you mentioned, so your survey that you did, it was global.
Yeah.
Did you see a difference between where the United States and Canada as compared to the UK and Europe and Australia? Is the US, North America, are we keeping up or are we a little bit behind? Because I know there's so much research that seems to come out of Europe with MPS.
Yeah, in terms, we're keeping up. Also, the big difference is the law part of it. The legal systems are different between countries. One country might be able to take to court something they do at a university lab versus in the US, we're not going to do that. So that I think might be the kind of biggest difference. But overall, globally, the field altogether seems to be in that same place of what are the big needs that people could use to implement it.
So people have either just gotten it or trying to figure it out, or they're trying to get funding and planning to get it going.
Yep, to convince their manager.
There's not a lot of laboratories that are doing it routinely. Certainly not, I wouldn't think government laboratories. I would think it's more likely in the private labs that would be able to get the funding for these. Maybe, no? Teresa's waving at me.
I did just want to say there is a publication out of the FBI where they validated the control region of mito with MPS for casework.
Wonderful.
Yeah, it's relatively new. That's why I was waving at you. I know of one government lab that has done that, but that's the only one that I'm aware of that has validated the mito for casework.
And I'm going to point out really quick, too, that we're not advocating any particular type of platform because there's a couple of different companies that make this instrumentation. Soft Genetics does software. You're not the only software company out there that does it. There's different tools and there's different software on the market. And that's part of, I think, laboratories are like, I don't know, which one do I buy? Which analysis software is the best one for me to use? And it's such a complex laboratory test. It's not only you have to get it and develop it internally and validate it and get all your procedures all in a line and understand how to do all the analysis on it. It's then I have to go and explain it in the courtroom. And so right now it's like that's a little bit further down the road, it seems.
Yes. Yeah. And that actually is one of our questions of when do you think this will actually be running at least like the crime scene type of samples. I think we'll see it with missing persons, that type of casework. That'll happen a lot faster.
Well, and I think we have seen it for that kind of casework. Yeah. It's definitely helpful right now in the identification of human remains for sure, because you can see a lot of like the National Center for Missing Persons. They've definitely made some good identifications through this type of technology. But using it on a blood stain at the crime scene and comparing that back to the reference sample of a persons of interest, it's a bit more complicated because it's much more expensive just to do one sample, let alone the crime scene sample and the reference samples for your suspects and your victims and everything. And the equipment itself, if I remember correctly, because I've bought the Verigen MySeq FGX, and I mean, there's some negotiating room on prices, but you're still looking at easily $250,000 to get one of these machines into your laboratory. And it's not something you can't just run like one or two samples at a time. You need to run, I would think, at least 24 samples to even break even on cost just to run it. So it's the advantage though is that they can run STRs, the new YSTRs, and the mito all in the same sample. So the cost benefit would be looking at, as Megan was saying earlier, multiplexing them, and with the standard methods, we need the CE for the autosomals, for the YSTRs, and then for the mito, so many different reactions, whereas with the MPS, they can all be done at the same time on the same sample. So that can help. So it can help bring down the cost. There was another article, I have to look for it, where they were saying that it was coming down in cost because of the chemistries becoming available. So again, this technology is developing, and getting things to the point where they're cost efficient is definitely a goal. I remember seeing a chart. You mentioned the Human Genome Project earlier. I remembered seeing a chart of what the cost was for sequencing way back at the beginning of that project, and it's $10,000 a sample or something. Something ridiculous. And then over time, that is just like a downward ski slope, because it goes right down, and like you can do a whole genome for $1,000 or something now. Yeah. So I guess depending on what the genome is. But yeah, you're right. So each individual, you're going to do your blood stain or your semen stain in a sexual assault case. The beginning part of it is the same. You still have to purify your sample. You still need to measure how much of your sample that you have. Then you decide on what kind of technology you want to use. And that's probably going to be something that labs will have to have a plan for, to understand, okay, so am I going to do my traditional STR analysis on, like Teresa mentioned, the capillary electrophoresis system? Or do I only have enough to do Y STRs? Or if I have this new technology available to me, can I do STRs, Y STRs, and mito all in the same thing? And it is definitely a cost benefit analysis, but it's not something that small laboratories would be able to do. You need to be a larger laboratory because you've got to have that economy of scale, I would think, to be able to have the purchasing power to get the supplies in the beginning. And these machines don't like to sit around doing nothing.
No, that's a really good point.
Oh, they get crap.
They like to be busy. So there's all these things that laboratories are struggling with. And I think maybe to relate this back to something for people who aren't actually in the laboratory, all of this technology is used very heavily in the biogeographical ancestry side of things. Now, there are definite forensic applications for that, but people who are sending their DNA, spitting it into a tube and sending their DNA through the mail, they are benefiting from these technologies, right?
Yeah. Same instruments. So that we
it's the same instruments, the same technology. They're using some fancy software to do their analyses, and that's how they're able to tell you that you're this percent African or sub-Saharan African, and this percent European, and this percent indigenous people. So that's where that comes from. It's this technology that's allowing that sort of thing to happen. So I wanted to switch more, and let's talk a little bit. We talked around the technology, what we're doing with it, and where we're at. So let's say we get up and running with this technology in a laboratory and we've generated all of this data. What does soft genetics do that can help us understand what we're looking at?
Sure. Well, with the MPS data, you need software to import it because we're literally looking at terabytes, which is a term that makes my head just kind of implode. Terabytes worth of sequence information. The software is based on a user interface where they bring in the data file and they have different settings depending on what their chemistry was. And if it's mito, it will align all of these sequences to a reference sequence. So there's a standard reference sequence that's used in forensics, and there are specific ways that it has always been lined up by this. When analyzing Sanger data, the software enables the analyst to look at the whole depth of coverage. So they aren't looking at just a single sequence, as we were saying earlier. They're looking at hundreds or thousands of each of these little fragments lined up. And that way they can very easily visualize any minor mutations, which would put that person in a different haplogroup. So that would be for the mito. And then for the STRs, as Megan was saying, the capillary electrophoresis, or the older method, they see more of a graph with, if there are 8 copies at that one particular chromosome, it's a peak at that location. With the MPS software, GeneMarker HTS, then it aligns these and looks down through the depth. And so if half of those 8s, maybe half came from mom and the other half, of course, from dad, so they're homozygous 8 for their length. But if one of their parents had a minor difference in that repeat section, that's detected. And so it's presented then in a bar graph and also in a table that shows what's the balance, how much of this 8 is the same. And so that's where you get this additional information is in looking, they call them isoalleles, because they have the same length. If I go back to, I love Ruden's analogy to the train, or you go back to that, the train is the same length, so that they have the same number of those repeats. But if half of them have a different repeat sequence in them, it shows up and you'd expect to see that in a balance, pretty much half and half. And so it has the ability then, when you're looking at mixtures, to be able to look at if your minor contributor maybe it has a mutation in it, it would show up as a separate 8 allele. So it's going to make it possible to tease out more information from mixtures.
I have to say, I think that's one of the things that got me most excited when I first started learning about MPS way back, was the ability for mixture deconvolution, the ability to discriminate between individuals. Because if you have, a lot of times in sexual assault cases, you've got biological relatives that are in the same mixture. And if you've got these isoalleles in there, that will help you be able to tell close relatives apart, and there was no way for us to do that with the traditional STR testing at all. And this is like the upholstery in Mom's train car had the blue seats, but their daughter has green seats or something. That's like we're talking about with the isoalleles, right? So the number of repeats are the same. It's just the stuff that's on the interior is slightly different. And it's those slight differences that this technology is letting us see.
Right. And so in the user interface, the bar graphs are nice because they're very similar to the appearance of the previous method. They call them electropherograms, those graphs we were talking about with the peak. Well, bar graph is very similar. So that's a nice visual. But then the table shows you the actual balance. Because if it's just a little bit of a minor, if it's single source sample and there's a little bit of a minor difference, they might have a mutation or it might be some noise. And that's where Megan's work on validation comes in. How much is noise?
Looking at these bars, and I remember they tend to be color coded, so it's a nice visual representation. So you can see there's this minor thing that's there, and that's maybe 10% of the mixture, and someone else is 90% on that allele. Are there ways for us to incorporate that into the statistical analysis as well, Megan? Is that something you're looking at too? You've got that percentage, the minor contributor and the major contributor there with the isoalleles. Since we had that information, it'd be nice, do we weight the statistic in some way to be able to help with a weight of the evidence that we find?
Yeah, absolutely. The probabilistic genotyping softwares that I mentioned will actually go in and look at the profile and look at a bunch of different factors about a DNA profile, way more advanced than I would want to get into, but it can look at patterns that are common to see and look at the percentages of people or percentages of the peaks that are there or those sequences and try to tease out different individuals and do what's called deconvolution. So that's a lot of research that we still need to be able to get the sequence data into the software. And right now, our basic using our regular STRs with the CE type of methods, we can do that because we're just looking at that number repeat that's there, looking at that percent intensity of the different contributors. But now, if you think every piece of DNA we had before, there's a bunch of different things we have to consider when it's sequenced. Right. So it's much more in detail, but we need to teach the probabilistic genotyping software these new patterns, the new details that we can see in sequence data.
Yeah, the details, I mean, a real basic one is that by capillary electrophoresis, a person can have one allele, the homozygous, the same, or they can be heterozygous. But with the deep sequencing, that person that's heterozygous, if they have isoalleles, they might have 4 alleles at one locus and still be a single source. So yeah, it's, as Megan said, a lot more information. That's just one little part of it that needs to be incorporated to be able to make use of all that information.
So it happens quite often. People will submit cases to the laboratory. They'll be like, it's a rush. I need it tomorrow.
Oh, no.
It's not going to happen with this technology.
No, it's definitely developing, but it is moving forward at a very good pace. We've been working with MPS sequence here with the research software at Soft Genetics since 2007, I think. And my husband said to me, he says, you got to learn this next gen stuff. You're going to apply that in forensic. I said, no. Forensic community has to move very methodically forward. I will be retired before they get there. And they've gone and made a liar out of me because it is definitely up and coming and it's moving. I think it's moving at a very good pace because it's very, just like forensic science, everything. It's very methodical because you're looking at something that is a legal application of science.
Just a general question. I think that this is especially true for public laboratories. They're overburdened. They've got more cases than they can handle. They've got long turnaround times. And implementing this type of technology isn't something that you can do overnight. It takes time. And I noticed that as laboratories started to adopt probabilistic genotyping as the statistical method that we try to use routinely for our standard STR casework that we do now, there have been some growing pains with that, where people have actually given incorrect testimony in court, cases have gotten overturned, partially related to mixture interpretation, differences in opinions between the state laboratories expert and maybe an external expert. So basically our old technology or a routine technology with a new statistical method, we've got issues in the field where people don't necessarily understand it as well as they need to in order to be properly applying it and explaining it in a courtroom. How do you see the MPS working out? Because I have my concerns because we are exponentially more complex. Not only in the laboratory method's a little bit more complicated, then you've got the data analysis, then we've got to present this data. What kind of issues do you guys see coming from that? And maybe this is more for Megan, since she's more forensic lab person.
Yeah, well, I was going to say education, but yeah, Megan, in terms of just validation process and making transitions, that's your area.
Yeah, the validation I did when I first validated the system we were using, and I was working in a research lab. It wasn't even a casework lab. And I did my first run, single source, repeatability. And I went, oh my gosh, there's a lot more we have to consider, because each component of what we do in analysis, you have that much more data. So I think some of the growing pains I think are going to be similar based on the survey that we did too. And just, it's the education, it's getting everyone on the same page of understanding. Now we have these ISO alleles. Are we even ready to do statistics even on those yet? We're familiar with a basic allele. So I think the validation process is going to take longer. And you're right. People, some of our survey respondents too said, I'm still trying to validate PropGen, probabilistic genotyping. I can't even fathom bringing on this whole new instrument platform. Luckily, the hands-on process, it can be automated, which is great. And it is stuff we're already used to as benchworking scientists with the types of pipetting and everything like that. But yeah, I do think getting to court, it's going to be the same hiccups, which hopefully some of the research that's being out there hopefully will help smooth some of these out so that labs are going in it together instead of just trying to deal with it by themselves, you know? Yeah.
So the education, it's definitely something that, like going to this workshop that's free at AFTA would be very beneficial for people. And then also, I would encourage anybody who's listening that's a legal professional, don't rely on the laboratory to keep you informed. You should take the time to try to go to these workshops and maybe talk to companies like Soft Genetics and get a little bit more idea, researchers like yourself, Megan, that are trying to work on it to get a better understanding of where the field's going, especially if you're somebody that's on a cold case integrity unit. This type of information might be good for you to have, especially if you're going through old evidence, trying to see, is there something new available that I might be able to do? Yeah, because I think it's funny. I'm remembering at the beginning of the podcast, Teresa, you mentioned RFLP, the original DNA fingerprinting. And I've been in court and had people, maybe even in the past 5 years, and the prosecution starts trying to introduce DNA evidence in the courtroom, and they're asking old questions from the RFLP days. So the technology's improving. Like you said, forensics is slow and methodical, but at the same time, we're still faster than our customers.
And another point of that is a lot of what Sarah and I and the other support people here do is just like online assistance and training. And so we've worked with public defenders and Project Innocence where they didn't do the wet lab production, but they have the data files. And so by putting them into the software, they can then visualize it also. So it's not limited. The software used is not limited to the laboratory, but to anyone who is needing to evaluate or use the sequence information.
That's really awesome. So that's a resource for people that non-lab people go to you and get some help.
Yes. Yep. So we can show them how to use the software. Yep. We currently don't do analysis as a service, but we do education so that if they want to be users of the software, they can learn how.
That's really amazing that you guys are able to help educate that way. We are at an hour already on our recording.
Isn't that fun?
So,
nerdy fun.
So did anybody have any closing remarks that they wanted to make? Sarah's shaking her head.
Sarah and I are like counterparts where they can't keep me quiet, and we have to encourage her to speak because you have this experience, Sarah.
One-on-one help, phone a friend.
Yeah, there you go. Exactly.
Sarah.
And
Okay.
Patience Unlimited. That's another one of her strengths.
That's a good virtue to have.
Absolutely. In having the workshop open to everybody, we're going to try to get a variety of different speakers. So some from universities, one from a lab in New Zealand, and just trying to give more of an overall picture. So because the technology is coming down the line at a nice pace, and it's something that is a resource for people for their continuing education. I mean, as they get it into the universities, people now at universities are trained in PCR and CE, and so this is going to be another layer added to that. But there's probably at least a 10-year gap between when it's part of the normal formal education process. So workshops are an excellent way. Resources from, like you said, the companies that make the software, are resources for helping companies that make the chemistries and the instruments are another resource.
It's really good that you're bringing in speakers from overseas as well, because it's a global thing that we're looking at, this technology. And like Megan said earlier, that we're all in the same boat here. We're all trying to figure out the same thing, and we're working together for that. And let's all learn from each other. Like someone else, I didn't work this way for me, so I had to try it this other way. Absolutely. And Megan, you're going to talk more about the survey results from your research projects.
Yep. And some solutions I at least know of right now as well. I try to give you those too. Yeah.
And remember, if you're not part of the solution, you're part of the precipitant.
So we are having a nerd conversation. You gotta love it. Gotta love it.
Okay. And on that nerdy note, thank you, ladies, very much for your time today. I appreciate it. And if anybody who didn't get that joke, you'll look up the word precipitant and solution, and it will make sense for you. But it's not funny anymore if you have to explain it. All right. So thank you very much, ladies. I'm going to get this edited and up as soon as I can. I hope to have this up before the workshop goes. But if anybody who's interested in attending the workshop, you can visit afdaa.org and click on meetings and workshops, and that will take you to information about the main meeting, the workshop, and links to register. But you'll need to register very quickly because it's coming up very fast. And if you want to make sure you have a cookie and a drink, then you need to RSVP for the meeting. With that, thank you, ladies, and I hope everyone has a great day.
Thank you.
Thank you.